some of you remember, a month ago we had this important immunologist, evolutionary immunologist from the Munster University, to be honored to have today Professor Larry Dishaw from US. Actually, we are now starting to work also in our laboratory in the biome, in the symbiosis, in the communication, the molecular mechanisms underlying communication between the microbiome and the hosts within the holobiome. So it's a great pleasure for us to have one of the most important members of the American community of comparative immunology, Professor Larry Dishaw. And it comes from another very important laboratory in the US, the one led by Emeritus Professor Gary Littman, and I remember also Adrian Weissman at Stanford University, also working on the immunology, on the innate immune system of invertebrates. So Professor Larry Dishaw is actually assistant professor at the University of South Florida in the College of Medicine, with appointments in the Department of Molecular Medicine and at the College of Marine Science. And he is also a core faculty member of the University of South Florida Genomics Institute. So he got the degree, graduated in biology at the Florida International University in Miami. All his life was in Florida for his career, with a thesis on the immunophylogenetic aspects of a Gorgonian coral. And as postdoc, then he moved to St. Petersburg in Florida, at the University of South Florida, indeed in the laboratory of Professor Gary Littman, where he started working on the evolution of the phylogeny of the new responses. All this text has been written by Dr. Susie Liberti here, who was also postdoc in the laboratory of Professor Larry Dishaw. So his research interests have always been to utilize comparative immunology approaches to help define the evolution of innate and adaptive immunity in the gut. And is interested in defining both conserved and novel elements in the innate immune dialogue between microbiome, resident microbiomal communities, and host organisms. And for this reason, his lab has been the first to develop a protocol-based model system to study this interaction, host bacterial interactions in the gut. And his leading actually is very focusing on the effort to demonstrate a role for gut immunity in supporting and modulating biofilms. And is also indeed looking at the therapeutic pathological aspects of these interactions in the human health. So it's a real pleasure today to listen about secreted immune effectors, how they shape the ecology of microbiomes, and how Ciona robusta tell us lots of new things about this. Thank you very much. Yes.
Thank you so much for having me. It's my pleasure to be here. Hopefully I don't do this when I talk, because I use my arms a lot. I'll try to keep my arm here so the microphone works. Yeah, it's a great pleasure to be here and share with you the work we've been doing in the lab for the past essentially 10 years now. This work is a culmination of different projects that it was hard to predict even 10 years ago where it would be today, because in this kind of model system, you follow the science, and the science takes you in new directions. And one thing led to another. We were initially interested in understanding just how, asking simple questions on how the gut immune system functions in Ciona, and trying to understand this new protein family that we had discovered, the BCPs, that I'll get into in a little while. And one thing leads to another, and we're working on, bacteria, the phages or the viruses that infect them, and then fungi. So my lab is doing this trans-kingdom approach now, looking at these different components of the gut microbiome and how they interact with host immunity. And my interest for a while now has really been on this idea that secreted immune effectors. And these are effectors or proteins or compounds that the immune system is secreting into the environment, that they function in unique ways to regulate the ecology of the outside world, and that this is really important for regulating how the host immune system behaves. Because the host immune system has two types of effectors, right? The effectors that are membrane bound, and the ones that are membrane bound, microbes bind to them, and deliver a signal to the cell that tells the cell to respond. But then the immune system also makes secreted effectors that are secreted into the environment. And those effectors are things that really interest me, because I wanted to understand, why would the immune system devote so much energy into making all this protein that it just dumps into the environment? And it's because these proteins are being dumped into the environment because they're doing something out there. They're serving a role in shaping how these microbial communities interact, how they settle, and how they interact with each other. So it's shaping the ecosystem of the environment. And that's really where my research has been going in the last few years, and it's really something that I've become really passionate about. So I'm going to back up, and before I even get into the stuff that we're working with in Ciona, I'm going to give you a little bit of background on the immune system, because some of you may not be too familiar with immunity, and I want to put our data in the context of mammalian immunity, so that you can understand how using a model system like Ciona can be relevant to diverse questions that are being asked, not just in simple model systems, but even in translational sciences, which is where the big money is, right, with human studies. So a little intro on the gut microbiome. It's a big trendy area that everybody's interested in now. An overview of the immune system. What are secreted immune effectors? I kind of just told you. Secreted antibodies, like IgA, that are secreted, and I'll give a little background on that. And then I'll jump into the current data that we have using Ciona as a model system. So the microbiome is this really hot topic now in the last 15 years or so, where people now know that the gut of animals, pretty much every surface of the animal, you've got skin microbiome, oropharynx microbiome, lung microbiome, vaginal microbiome, gut microbiome. So like all these mucosal spaces and ectodermally derived or ectodermally coated, epithelial coated tissues are all these surfaces are colonized and coated essentially by this complex community of microbes. However, in the last 15 years, the primary focus of microbiome sciences has always been to sequence what's there and to characterize what's there, but the low-hanging fruit has always been bacteria. So most of what we know is bacteria, which is for good reason. Bacteria contribute the most number of gene products, they contribute the most to the physiology of the animal, because there's so many metabolic activities that bacteria can do, and they're so abundant and diverse in these microbiomes. But we wouldn't have known that without sequencing them first. But we know that microbiomes are consistent of not just bacteria, but the phages and the viruses that infect them, right? Fungi, protozoans. Protozoans make a big part. Ciliated protozoans like to feed on bacterial communities. They also they graze and they feed on mucus. And then helminthic worms. So the microbiome is really this complex microcosm of ecological reaction that's kind of like soil, except in your body. And the gut is this great environment to ask these questions of how microbial communities interact with each other, because the gut is not just a confined space, but the gut is a very well-adapted organ for removing water from the system. So the gut of animals removes water from the system, and in doing so, it reduces the volume and it brings things into proximity. And proximity is key to microbial interactions with each other. And that's important because microbes interact with each other more than they interact with the host. And oftentimes the microbes are just on the host hitching a ride, finding a home that's rich with nutrients, or a home that's a safe space. But they're usually, most microbes that colonize animals are not pathogenic at all. And that's what we're finding, is that this is important because pathogens cause problems, but pathogens are pretty rare in the environment. Most people estimate less than 1% of all the microbes ever trying to cause infection. And the amount, especially like a filter feeder, this is why an animal like Ciona interests me, because a filter feeder is filtering all this water that's microbe-rich all day long, 24 hours a day, 7 days a week. And how does its immune system deal with that? And how does it not respond to everything? And those are important questions, right? Like, why does the immune system tolerate being coated in microbes and covered in microbes? And that's because the pathogens behave differently. They're rare and they behave differently. The pathogens often are trying to penetrate, you know? And we'll talk about that a little bit later. But the microbiome has really become relevant and important to most studies because we know now that the microbiome appears, like the human microbiome appears to affect physiology in all parts of the body. Okay, and it's established really early on in infancy. Okay, the moment you're born, you're being colonized by microbes. And the microbiome experiences a temporal aspect as well, right? There's components of the microbiomes that are affected by hours and days, right? And the microbiome even has a diurnal cycle, where the microbiome is affected by light-dark cycles. And then diet, right? And time in general changes the microbiome, you know, as the animal ages. Their immune system changes, their diet changes, their behavior changes, that affects the microbiome. So the microbiome is affected by things that occur within the hours or things that occur within the lifespan of the organism. And this is important because if the microbiome is shaping our physiology, understanding how that microbiome changes over time is really important. And so, but part of our understanding of all these things about the microbiome is really to understand how the host immune system modulates this ecosystem of microbes in a way, I believe, that the host immune system is serving critical functions into helping preserve the stability of the microbiome. Because the microbiome has been co-evolving with animals for eons, right? Hundreds of millions of years, most likely. Probably since the origin of animals, they've been colonized by microbes. And because of that, especially animals that develop externally, they are dependent, they become interdependent on microbial interactions. So the immune system can be viewed almost as an interface that helps to govern interactions with the microbial communities of the environment that help benefit your physiology. So I kind of jumped ahead, but how is the microbiome typically studied? Most people are sequencing, which is really important to understand what's there, especially to understand what's there over time. And then a lot of people are doing now metabolomics, which is studies, and metabolomics uses mostly like urine, but you can now use fecal material and blood to study the metabolites that come from the microbiome. So right now, at latest count, we know that the microbiome is shaping physiology, not just by helping you process calories, but metabolites and metabolic products from the microbiome get absorbed by the epithelium, get into the bloodstream, and influence physiology, right? And there's probably 200 metabolites right now that are recognized as coming from the microbiome. So very exciting. But we and others are also trying to understand the biology and ecology of the microbiome by culturing as many of these microbes as we can to do in vitro experiments to understand how the microbiome basically exists and how components of the microbiomes grow and how they behave. And this is this new developing field which is basically called, culturomics. The idea that we're culturing a lot of things to understand their interactions, and then sequencing, because the things that you're culturing, you can also sequence those in vitro interactions and study the metabolites being produced as well. Because often when you have polymicrobial growth, polymicrobial biofilms, those interactions are generating products that they wouldn't make on their own. So if you grow bacteria X by itself, or bacteria Y by itself, they make two different products. But if you grow X and Y together, they make unique products because they're interacting with each other. So doing co-culture is a really important part of understanding how the microbiome is functioning. And I already mentioned that the gut is this great microcosm of confined space, forcing things together to interact. And when you're doing polymicrobial interaction studies, proximity, recognizing that proximity is important, is key to the experiments. And often people talk about the microbiome and we sequence it, all this stuff from fecal material in humans, but an advantage of an animal like Ciona is that you can go in there and pull the gut out and actually sequence right from the gut, including material that doesn't come out with the fecal material, because the fecal material gives you just a glimpse of what's there. But the microbiome essentially is living as these polymicrobial communities at the surface of the epithelium. So here's an example of epithelial cells, and this right here would be the mucus layer, and all that staining is these rich polymicrobial communities basically colonizing the mucus as biofilms. So at the front lines of these interactions, of course, are epithelial-based defenses. And the epithelium is the first line of defense, and the epithelium, most people don't recognize that the epithelium itself is a fully functional immune system on its own. The epithelium in humans even expresses MHC, so the epithelium has all kinds of capacities to do a lot for the immune system. And in most cases, the epithelial cells are actually polarized, so that you can have expression of proteins on the outside of the animal, and a unique set of expression on the inside of the animal, which is really interesting, that polarized expression, because there's toll-like receptors that are expressed on the inside of the animal, and those toll-like receptors would provide danger signals if bacteria get through, because normally those toll-like receptors are never engaged on the inside of the epithelium, and if bacteria get through and they get engaged, that's a direct danger signal. So, just to put it into context what we're doing, I just want to give a little background on the adaptive immune system is different than the innate immune system, because the adaptive immune system, right, involves the rearrangement of germline genes to make new receptor combinations, and then you get antigen-specific responses in somatic lineages. And invertebrates generally don't have this. It's always been recognized that this is a vertebrate invention, okay? But only recently are we recognizing that maybe there's exceptions to the rule. Like in the sea urchin, they're rearranging this class of transformer genes. But this is all very new stuff. But historically, the idea that adaptive immunity is unique to vertebrates is kind of like the mainstream immunology thinking. And one way to think about this is that clonal selection of B cells, for example, if you have a lot of different B cells and you have a pathogen, one B cell recognizes that pathogen. All these B cells are expressing unique repertoires of receptors, the B cell receptor. One of them recognizes the pathogen and it expands, and you get specific clonal expansion of somatic lineages that are specific to that antigen. And only the adaptive immune system is known to do this, right? And then innate immunity is different. Innate immunity, you have a lot of population level diversity of alleles. And then a pathogen is recognized by a single Ciona, let's say, that can recognize this pathogen, will survive and expand, and its progeny now is resistant to that new pathogen. So that's how kind of innate immunity works. And adaptive immunity, vertebrates do the same thing, because adaptive immunity in most vertebrates doesn't develop for the first few years of life. So in the first few years of life, you have a lot of this going on, right? You have selection by the innate immune system, where a lot of childhood illnesses that kill babies really young. This is happening. You're selecting for these resistant clones. Of course, now with medical interventions, we bypass evolution and we vaccinate kids and stuff. But this is a very busy slide, I understand. I just wanted to give you an example of the epithelial interface. And IgA is an antibody that the adaptive immune system of vertebrates makes, right? And IgA is an antibody that's secreted into mucosal fluids. And IgA has a variety of functions that include immune exclusion, which means that the IgA is secreted outside and binds bacteria. It keeps bacteria from adhering, and they basically go out as poop, right? So that immune exclusion, it keeps microbes from binding to the epithelium. But IgA-bound microbes can also induce tolerance by delivering tolerogenic signals. If they bind the right cells, when they bind M cells, they can send signals to the dendritic cells, which can induce tolerance. So, and then there's also signals that are delivered that can help maintain homeostasis. So IgA has a lot of different functions that are not just secreting antibody into the luminal space to stimulate immune exclusion. But the reason I wanted to talk about this is that IgA is doing this, but this effect is not unique to IgA, because IgA is something that mammals and birds have, but amphibians make IgX, and fish make IgT, for example, and they're not even homologous. So these are de novo antibodies that have evolved in these lineages to serve very similar functions. IgT in fish is doing the very same thing that IgA does in mammals and birds, and IgX in amphibians. And the reason I bring this up is that in Ciona, this VCP, which is an Ig-like protein, seems to have functions that are reminiscent of what IgA, IgT, IgX does. And that would suggest that chordates in general evolved these secretory antibodies into mucosal tissues as a way to interact with the microbiome, maybe. That the production of immunoglobulin-like proteins and secreting them into mucosal environments is something that chordates are likely have become dependent on, because it seems like all these different chordates have some kind of Ig-like protein that they secrete into the environment. You know, like mammals secrete tons of it, 3 grams a day are being secreted. That's a lot of energy to invest into protein that you're going to poop out. This is what fascinates me. Why are these secreted immune effectors? We make so much of them. Why do we invest so much energy if they're just leaving the body? And that's because when they're secreted, they're serving critical functions and shaping the ecology of the system. And those are the kinds of things that really interest me. In the adaptive immune system, you have a difference between natural antibody and antigen-specific. So the natural antibody is more like the innate immune systems. The natural antibody is stuff that mom secretes in the breast milk. The natural antibody tends to have low affinity, high diverse antigen recognition. So like antibody against LPS would be considered a natural antibody because it will recognize all gram-negative bacteria. So antigen-specific is when an antibody binds some kind of bacteria or a pathogen that's a threat, it's internalized and recognized by specific B cells that then make clonal antibody that are specific to that bacteria. And in the future, on a second encounter, it can neutralize that pathogen. But most of the antibody that circulates in our body for the first 3 years of our life is just natural antibody. And natural antibody we get from mom in utero, and mom delivers through nursing. And natural antibody protects the baby against a wide range of microbes. The rest of it's not very important. We'll jump in. So secreted immune effectors shape the ecology of microbiomes. This is something that really fascinates me, right? So these secreted effectors include all kinds of stuff, right? Antimicrobial peptides, like REG3 and defensins, C-type lectins, which include mannose-binding lectin. And we know that a ton of that's being made in the gut as well. Antibodies, of course, like IgA and mother's milk can supply IgA. Lactoferrin is something that's very rich in mother's milk as well. Lactoferrin is another type of antimicrobial peptide. These are things that are produced in grams by animals and being secreted into the environment. Why would an animal invest so much energy in this stuff? Okay? The answer could be where do these microbes live? And the biofilms that are forming on the surface of the epithelial cells, because the bacteria that grow as stationary growth, you know, when we grow bacteria in a test tube in the lab, that's an artifact, right? They never grow like this in the environment, vortexing in a liquid culture. Bacteria in the environment tend to adhere to some kind of surface. Even bacteria that we used to think were planktonic in the ocean, if you look carefully enough, they're actually attached to a microparticle of something, some type of carbon-rich substrate, some type of carbon detritus or debris. They're always attached to something, even if it's a piece of mucus from a fish, a shed from animals. Mucus floating around that you never even see it will be home for microbial growth. So microbes like to adhere to things and form biofilms. And when they form a biofilm, they start secreting polysaccharides, and that polysaccharide allows them to grow as an adherent community, where in that biofilm they're protected from the environment, but they can interact with each other, and you can have biofilms of single bacteria or biofilms of multi-polymicrobial communities. And are you guys familiar with quorum sensing, which are when bacteria secrete small molecules to interact with each other? That occurs only in biofilms, right? Quorum sensing is a biofilm phenomenon because in suspension, if a bacteria secreted any kind of quorum sensing molecule, it would just disappear, it would just float away. So in order for the quorum sensing molecule to relay information so they can communicate, it has to occur inside of a matrix. And this, I think, is key to understanding how the microbiome functions. Okay, so this is, unfortunately, you can't see it too well, but the blue here is epithelium, and this acridine orange stained material is biofilms growing in the mucus, right? And this came from work of my colleague, Bill Parker at Duke, and he first discovered this 20 years ago, that there's all kinds of biofilms in the gut of animals that are adherent to the mucus, and most of these biofilms are richly coated with IgA. I'll get to that in a minute. And at the time when he first proposed his ideas, everybody thought he was crazy. And he tried unsuccessfully for many years to get NIH funding. And they thought it was a wild idea that the immune system would be working in collaboration with microbes. Why would the immune system do that? You know? But in those other pictures that I showed of the microbiome that here, you saw the epithelium, and then you see a black space between the epithelium and the overlaying microbe-rich mucus. That black space is not an artifact of histology. If you were to actually stain it with crystal violet or Kumasi or acridine orange, you would see that the mucus goes all the way to the epithelium. But they were stained for microbes, and it leaves a black space. And that black space, which is here, this is microbes out here. That black space is a space that is created by antimicrobial peptides that are secreted and collect. And so this is an example of a Reg3 knockout. The microbes then get across. So the antimicrobial peptides often collect in the dense areas of the mucus that are adherent to the surface of the epithelium, and that helps create a space between the microbiome and the host. And a lot of antimicrobial peptides are bactericidal, and they can often lyse bacteria, they can neutralize bacteria. So the mucus collects antimicrobial peptides as a way to create space with the microbiome. And here's an example of another epithelial surface. The inner mucus space is usually devoid of microbes, okay? And the outer mucus space is IgA positive, and that's what that signal is, IgA. So IgA localized into the outer mucus layers is really relevant to what we see in Ciona with the localization of VCPs. The secretory component of IgA helps it localize to the mucus. The inner mucus layer is devoid of microbes, and products that are shed by the microbes can get across still and shape responses, and that's a whole other line of research. But it also promotes generating more IgA. But this is really important because 20 years ago, when Bill Parker discovered these biofilms that were rich in IgA, he started doing experiments in vitro that showed IgA can actually contribute to biofilm formation. And it was basically dependent, right? IgA, he didn't see it with any other antibody, and all the controls were negative. It wasn't a protein effect. It was concentration dependent, and you can heat deactivate the IgA and miss the response and neutralize the response. So it was a specific property of IgA to facilitate the formation of biofilms in ways that help encourage certain microbes to colonize, what he was proposing. 20 years ago, it was such a novel concept. Nobody was doing microbiomes, and everybody thought he was really a lone wolf with this idea. Because historically, when we do immunology experiments, antigen-antibody interactions, an antibody binds a bacteria and often induces agglutination. The agglutination response, you can see it under the microscope as granular material. In 99% of immunology experiments, when you do an agglutination response, that's your endpoint. The experiment's done and you go home. Parker asked, what happens if we wait? Because we know that when IgA agglutinates bacteria, they don't die. The IgA is not killing the bacteria. The IgA is not bactericidal. So he waited. And the next day, after there was agglutination, biofilm started spreading. So he proposed that bacteria in the gut become agglutinated by IgA, and that forces them to drop onto the mucus. And when they drop, they start forming biofilms. That's how they colonize. So this is an example, of his images, and he showed this in, rat, human appendix, and then I'll show you in a minute in rabbits. So this is IgA positive mucus, and all that co-stains with biofilms. And this is a full intestinal compartment from a rabbit, and he was able to show that all these compartments of the hindgut, and then here is biofilm negative. All these compartments of the hindgut had biofilm-rich IgA coating those surfaces of the hindgut. And he was convinced that IgA was necessary for colonization to occur in the gut. Lo and behold, 20 years later, this just came out this year, Mazmanian publishes a paper showing this made the cover of Science, like it was new finding. But it was just basically validating what Parker saw 20 years ago. That microbiota of the gut utilized IgA for mucosal colonization. And he was showing that some bacteria even modulate their surface architecture to invite IgA to bind. And that the IgA response was required for B. fragilis, at least, and other commensal species to occupy defined niches. So that when they created these knockout bacteria that couldn't invite the IgA binding, or they were neutralized IgA in this paper, these mice wouldn't colonize properly in their gut. Of course, this made Parker really angry. So Parker had to leave he left the IgA work years ago because he couldn't get it funded, and now he works with helminthic worms. So he's been working with the role of helminthic worms in interaction with the immune system. And now he wishes he was still on the IgA story. But anyway, in 2012, I contacted Parker, and this is how we wrote this review paper together, because I basically said, hey, I'm seeing something very similar to what you saw in this C-squirt that I work with. And these proteins that we discovered, these VCP proteins, are doing very similar things to what you saw. And he and I started basically talking, designing experiments, collaborating, and we ended up writing this paper as a review, but it's also a hypothesis paper because we talk about how we really think that these interactions between host effector molecules and binding the microbiome components are really this ancient property of how the immune system interacts with the environment. And it makes sense, right, to think that animals would evolve ways to tolerate non-pathogenic biofilms, because a biofilm from a non-pathogen, right, from a friendly bacteria growing on the surface of the epithelium, actually protects the animal from a pathogen. A pathogen cannot colonize if it has biofilms growing of friendly bacteria, right? Because the friendly bacteria would go to war with those pathogens, right? So in the ecology of the ecosystem, if you think about it that way, the ecology of the microbiome is basically microbes are constantly interacting with each other in ways that facilitate or protect the host immune system, keep the host immune system at bay, because the host shouldn't have to respond to most things if most things are non-pathogens, right? And microbes are typically too busy interacting with themselves or against each other, and they don't really care about us. They're just kind of hitching a ride, right? And getting nutrients from our diet and all that stuff. So what can we learn from Ciona, right? So now I finally missed Ciona. Hope that's in time. What can we learn from Ciona? Ciona, of course, has no adaptive immunity. We know this stuff. But the thing that really interested me when I first started working with Ciona, we were looking at BCPs with the Rosarius, the Santos and Pinto, and this is how Susie got recruited to the lab. We were really trying to understand BCPs in Ciona, and I basically, as a filter feeding organism, I asked this question, you know, does this animal have a microbiome? Because every animal has a microbiome. Is it a specific microbiome, or do microbes just kind of go through them all the time? And when we looked at the microbiome of Ciona from the Mediterranean, Woods Hole, and San Diego, even though these robusta and intestinalis are actually speciating now, and they're diverging, there is about 35 bacteria, which represents 13 unique families of bacteria that are absolutely conserved among San Diego, Woods Hole, and Mediterranean animals. And the animals that we got from the Mediterranean came from Fusaro Lake, which is as isolated as you can get. And yet the same microbes are there that you see in San Diego, those 35 bacteria that represent 13 different families, right? And these 13 families of bacteria represent this huge range of metabolic capabilities. And we also did the virome, and the virome shows even some evidence of compartmentalization, where the virome itself suggests that some of the bacteria live in confined niches, stomach, midgut, and hindgut, because we did the virome of the different compartments separately, and we could see that the virome, which consists mostly of phages, are phages that are against specific bacteria of the microbiome that appear to live in the same compartments. So this is really interesting that there's even evidence of some compartmentalization. But we like Ciona for obvious reasons, right? Because they're transparent, and you can see their gut. We can colonize them, of course, with fluorescent bacteria and watch where the bacteria colonize. And we can now grow the animals germ-free. Of course, it's not a germ-free mice. We don't grow them indefinitely, but we grow them for defined times, 4 to 6 weeks. If you're really ambitious, you can go longer. But we do all of our experiments in a short period of time, and we don't need to go that long, right? Because we're asking questions about how do we colonize a sterile animal in the first few weeks of life when their gut is first developing and their gut is first normally seeing microbes from the environment. So one of the things that we like from Ciona, of course, is that we can basically generate a dish full of these sterile animals, and this dish is basically little vertical guts that you can study it. And we, you know, Suzu even used to use these dish with glass cover slips in the bottom. You can go right to the microscope and get really nice images of these animals. So this is when we did the microbiome, which is looking at bacteria, and then we did 2 papers looking at the phages. And in fact, this recent paper where we characterized the small single-stranded DNA phages actually provides a tremendous amount of evidence that the Ciona gut is rich in endosymbiotic bacteria. And that's another line of work that we're going to try to pursue eventually. But there's evidence from single-stranded phages that Ciona must be colonized by a diverse abundance of endosymbiotic bacteria, because we have a ton of evidence for these phages that are known to colonize endosymbiotic bacteria, which is really cool. In fact, we found 273 unique single-stranded DNA phage families in Ciona, and the human microbiome only has about 33. So Ciona seems to have this rich collection of these small viruses. But the most important part of the story is that when we did the virome, we found that at least half of the bacteria appear to be temperate phages, bacteria. Sorry, at least half of the bacteria appear to be lysogens because they're carrying prophages. So when we sequenced the virome, we found that most of the viruses that we were seeing were phages, of course, and most of the phages that we were seeing were temperate. And phages come in 2 flavors, right? Phages are bacteria. Viruses that infect bacteria, they come in 2 flavors. One flavor is a lytic phage. They recognize bacteria, and they lyse them, and when they generate more particles, the particles are released, kind of like your standard viral infection. Temperate phages infect bacteria and make a decision. They either go lytic or they integrate into the genome. And when they integrate into the genome, they integrate as a prophage, the prophage is essentially dormant, and that prophage makes the bacteria now lysogen. And a lysogen is a bacteria that carries phages in the genome. This is important, we think, because these phages appear to be shaping the